Hello, everyone. I'm Cedric from MDPI, and I will be your host for today. Welcome to the second live session of the first International Electronic Conference on Antibiotics, the equal power of antibiotics and antimicrobial resistance. This e-conference is organized by MDPI and the journal Antibiotics and is chaired by Professor John Gustafson from the Oklahoma State University in the United States. I would like to thank the conference chair, the conference committee, and the session chairs for making this e-conference possible, along with the speakers and, of course, our participants. The conference is now open for discussions, so until the 17th of May, you are able to read and comment all submissions presented to the conference. Please check the conference website to find the whole list of accepted submissions. I would like to remind everyone that this session is recorded. The recording will be made available as soon as possible and will be posted online alongside the first session from yesterday and the upcoming live sessions of this e-conference. This webinar is also being live streamed on MDPI's YouTube channel. Hello to those of you watching us on there. Today's live session is chaired by Dr. Marta Pérez Lloret from the School of Chemistry at the National University of Ireland and Dr. Anthony William Coleman from the Laboratoire des Multimatériaux et Interfaces in Villeurbanne, France. Thank you so much to both of you for chairing this session. There will be 4 presentations today. Each presentation will be followed by a question time moderated by our chairs. If you have any questions, please feel free to post them in the Q&A box, which you can find in the Zoom menu bar at the bottom of your screen. It would be great if you could use the Q&A box, not the chat panel. And if you wish to ask your question in person, please raise your virtual hand and we'll hand you the microphone. And that's it from me. Now it's time that I hand over the microphone to our 2 session chairs. Thank you and enjoy, everyone. Dr. Pérez Lloret and Dr. Coleman, the floor is yours.
Good afternoon, everybody. You have the benefit of 4 excellent speakers this afternoon. Yves Briers, Sara Soto, excuse me, please, Kim Lewis, and finally, James Collins. That's a great news for you. The bad news is that you have me and Marta as your co-chairs. So we're going to split this up. Marta will look after Yves Briers and Sara Soto, and then I will look after our American colleagues. As Cedric said, please use the quick Q&A or the hands up sign, whichever you want. Try not to use anything else to try and communicate, especially the chat. The time is limited, so I ask the speakers to get on with it. I will shut up very quickly and make your questions succinct because the Q&A system does not allow discussion. So I will now pass over the chair to Marta to take the first 2 sessions and take us through for the next hour and a bit. Okay, thank you.
Thanks, Tony, for the introduction. So today I'm honored to be chairing this session, second session of the International Electronic Conference on Antibiotics. Our first speaker, it's Yves Briers. To introduce him, I will talk a little bit about his life in the research field. So he obtained his PhD in bioscience engineer from the Catholic University of Leuven in Belgium. He then moved to Switzerland at the ETH Zurich with a long-term fellowship from the European Molecular Biology Organization. And he continued his postdoctoral path back in the Catholic University of Leuven. After, in 2015, he started as an assistant professor in Ghent University to finally become, in 2020, an associate professor. He is currently the head of the Laboratoire of Applied Biotechnology, and where he's leading his group too. With more than 100 publications and multiple patents, his research focuses on the synthetic biology of modular proteins, which can be applied for antimicrobial purposes, as probably he's going to explain to us soon. So let's give the spot to him and enjoy the talk.
All right. Thank you very much, Dr. Pérez Lloret, for the kind introduction. And hello, everyone. It's my pleasure to speak today for this audience of antibiotic experts. And I'm also looking forward to the next talk. I'm just the start of the session. I will speak about phage-encoded lysins as a broad class of antibacterials. And I will particularly focus on how we can engineer them to make them working in the most effective way. And we are working with enzymes. When you think about antibiotic, I'm sure most of you think about the small chemical molecules, the most traditional molecules, antibiotics which are now on the market and approved for clinical use are this kind of molecules. Some other people work on peptides or bacteriophages. There are even some other classes, but enzymes is for sure a very unusual group of molecules that's considered for use as an antibacterial. But I hope I will convince you in my talk that enzymes have a very promising mode of action as well. I will be focusing on enzymes that are produced by bacteriophages, or bacterial viruses. And in this cartoon, you see the replication cycle of a phage. And at the very end of a replication cycle, there is a lysis phase. So the phages have injected their genome, new phage particles have been produced inside the cell, they have been maturated, and at the very end, the phage produces an endolysin that will degrade the infected cell from the inside. And specific function is that it will degrade the peptide glycan. And the peptide glycan is the exoskeleton of a bacterium, and it keeps the shape of the cell intact. And this protein will be secreted, will pass in the membrane, and in the periplasm, it will degrade the peptide glycan, and it will result in osmotic lysis. This you can see in this electron microscopy image, all these white particles are phage particles, and this is truly a snapshot of the movement of lysis. You see that the cell is exploding, and that there are some remnants of the cell wall. And this explosion is important. It's due to the internal osmotic pressure, because thanks to this pressure, the new phage particles are disseminated so that they can look for new hosts to infect. This you can even see better on this little movie. You see here some cockle cells which are infected by a phage that also produces GFP. And what you see is phage lysis, so the cells explode, and in the end it's like a firework, and all the cells explode and disseminate the phages. So this is the lytic step of the replication cycle of phages. That's not the only phage lysin. There is a second stage in the cycle where bacteriophages have to pass the cell wall, and that's during injection of the genome. Prior to injection of the genome, the phage has to make a little hole in the peptide glycan to make space to inject the genome. And also for that step, the phage has a second lysin. It's called the virion-associated lysin that degrades locally the cell wall. But this lysin is controlled. It's linked, physically linked to the virion particle so that the hole remains small, so that the cell remains intact for the phage to be infected. So 2 lysins per phage, 1 endolysin and 1 virion-associated lysin. All right. These phage lysins are in fact an umbrella term. In fact, they cover many different enzymatic specificities. And so here you see the peptide glycan structure. You recognize the glycan chain, the peptides, and the cross links or the cross bridges in the case of gram-positive bacteria. And there are lysins that cut in the main chain, in the glycan chain. A major group are the red scissors, the myramidases or the transglycosylases, or the glucosaminidase that cut here, but many other endolysins or phage lysins in general, they cut in the peptide. So biochemically, it's a different enzymatic specificity, but the biological outcome for the phage is the same. The peptide, the glycan meshwork is disintegrated and there is osmotic lysis of the cell. So I just want to show you that there's a huge diversity at the biochemical level for the group of phage lysins. Also structurally, there is a large diversity of endolysins. We have so-called simple lysins, globular lysins, that have just a single domain, which is the enzymatic active domain. We call that the EAD, the enzymatically active domain. But the majority of lysins have a modular structure, and they have more than one domain. Besides an enzymatically active domain, they also have a cell wall binding domain that's needed to attach and bind to the cell wall prior to the cutting action by the enzymatic part. That's the CWD. And this is just a simple modular lysin, but there are many more lysins that comprises two EADs or that comprise two CWDs. So there's a large diversity at both the biochemical and structural level in the phage lysin group. We have recently collated all available information in the UniProt database linked to phage lysins. So 11,800 lysins are here brought together in a single database. You can browse the database, you can click on every link and you get a sheet with all the information retrieved from more than 10 different databases. And this database is a tool for the phage lysin community to investigate phage lysins. You can also use the Biomart where you can filter the entries in the database and you can export all the data as a sequence file so that you can do your own analysis. We have used our FALP database to visualize how diverse the phage lysins are. What you see in this image is on the y-axis, you have all kinds of genera of bacteria, gram-positive, gram-negative, and on the x-axis, you see all the CBDs, the cell wall binding domains, and the EADs, the enzymatically active domains. And this is just a little zoom in, and you see that phage lysins are truly a mosaic of domains driven by horizontal transfer. So for every phage has evolved its own optimized phage lysin to fulfill its biological function, being getting inside the cell and out of the cell. Here's another visualization that shows how complex or how diverse phage lysins can be. You see on this circle in blue all the enzymatically active domains. In red, you see all the cell wall binding domains, and all the lines indicate how these domains, how these building blocks can be connected. And you see it's a whole meshwork. There's a high diversity of combination of these building blocks can be made. You have to consider this as molecular Lego. And there are a lot of building blocks, and nature has designed the optimized phage lysins for its biological function. So conclusion, first take-home message is that phage lysins are a very diverse group of proteins. Okay. So why I am here in this conference on antibiotics? And that's because since about 2 decades, specifically since a paper published by the Fischetti Lab in 2001, that phage lysins are increasingly considered as antimicrobials. So what happens? So if you clone your lysin and you purify a recombinant form of the lysin and you add it to a gram-positive cell, what will happen is that it starts degrading the peptide glycan layers from the outside until the cell can not longer resist the internal pressure. And that's what you see here. The endolysin has been added and it punctures a hole in the cell wall, and due to the osmotic pressure inside the cell, the cytosol, the cytoplasm is extruded, and I think you believe me when I claim that this cell is dead immediately. These lysins work very quick. They usually work upon contact. It takes a second or 2 seconds or a few minutes to be active. So that's one of the benefits of enzymes. They are active molecules, they have a turnover rate, and they quickly act. It's a differentiating feature compared to many other antibiotics. They are specific, usually at the species level, sometimes at the genus level, or sometimes at the cellular level, but usually it's at the species level. So they're narrow spectrum antibiotics, and they have the benefits of those kind of antibiotics. Third feature is that they're highly bactericidal. So they kill the cells very quickly in a specific way with a high log reduction. So they work pretty well in vitro. This research has been picked up by companies, and the first clinical trials have been completed. And there are few licenses evaluated in the phase 1 and a phase 2 trial, and no adverse effects or clinical abnormalities have been reported so far. The single completed phase 2 trial has shown very encouraging results because they discovered the superior activity compared to standard of care antibiotics against MRSA in the phase 2 trial. And they did it for endocarditis and for bloodstream infections. This is not the easy kind of infections like skin infection. No, this is really the hard to treat kind of infections where they could show superior activity. And currently a phase 3 trial is going on. So I think I could convince you with this that phage ligands are for these gram-positives a very promising, encouraging, emerging class of antibacterials with a novel mode of action. Okay, but your antibiotic experts, you will likely know that if you go to this top 10 lists of the most urgent threats or serious threats or concerning threats, that it are not the gram-positive pathogens which are populating this list the most, but the gram-negative. If you look to this list of the CDC report published 2 years ago, you see Acinetobacter, Enterobacteriaceae, Campylobacter, and so on, Pseudomonas. There is much more need for novel antibiotics for gram-negatives. And phage ligands work easily on gram-positive pathogens, but what happens with the gram-negative? You know that gram-negative cells have an outer membrane, and this outer membrane is shielding off a lot of antibiotics that are active against gram-positives. And this is because the hydrophobic and hydrophilic barrier. Only the antibiotics which are small enough can pass through the porins. A lysin is 15 to 20, sometimes 60 kilodalton. Of course, it's much too large to get through this outer membrane. So the gram-negative bacteria are intrinsically resistant against lysins. And that was our playground in the last 15 years. We have investigated, we not alone, also other people, different strategies to make these phage ligands working against gram-negatives. And I will wrap up these 5 main strategies to give you an overview on the possibilities and opportunities. Okay, the first group are use of outer membrane permeabilizers along with the phage ligands. So the outer membrane is stabilized by calcium and magnesium ions. And you can use chelators like EDTA and citric acid that chelate these calcium or magnesium ions and thus destabilize the outer membrane. Another way in doing this is using polycationic compounds. What they will do is they will competitively displace the calcium and magnesium out of the outer membrane, thus also permeabilizing the outer membrane. And indeed, it works. So what you see on the left is a situation we use the CBD, a cell wall binding domain of a phage ligand, fused to GFP, and when you combine that with EDTA, that permeabilizes the outer membrane, you see that this protein gives a kind of jacket, a green jacket to the cells. So this means that this protein that normally cannot pass the outer membrane can get through the outer membrane and bind the cell wall. And when we did killing assays of the lysin EL188 together with EDTA, you see a dose-dependent effect and that you can kill the cells. So outer membrane permeabilizers can be used along with lysins to kill the cells. but they cannot easily be used in human medicine. EDTA is an anticoagulant. It's not something that you can use to treat a systemic infection. So outer membrane perolysis are nice, but cannot be used to treat human or veterinary infections. Another application, another strategy that we have demonstrated is the use of high hydrostatic pressure. High hydrostatic pressure is a technology which is used in food conservation. Food like oysters and guacamole, which cannot be pasteurized, need an alternative strategy to make it sterile. And that's where high hydrostatic pressure is investigated. So with high hydrostatic pressure, you can eliminate bacteria from this kind of food. But the equipment you need for that is very expensive. And it becomes more expensive when you need a higher pressure. So we tested if we could combine the high hydrostatic pressure along with the phage lysin to reduce the pressure needed. And this is what you see in this graph. When you applied high hydrostatic pressure, again with this fluorescent indicator protein, you can see that the pressure pushes the protein through the outer membrane and can bind the cells. So if you do the experiment again with the same phage lysin, with different pressures, you see that you can kill the cells as well. So with pressure alone, you have the gray bar between 1 and 2 log. But if you combine it with an enzyme, you can increase the killing rate. So the pressure also pushes the lysin through the outer membrane. So this is a nice application for food conservation, but again, it's not something you will use in human or veterinary medicine. And you cannot put a patient in a machine and apply 200 millipascal. Okay. That brings me to the third strategy, and that's the main strategy I will also discuss today. That are the RT lysins. Here we used protein engineering. So we engineered the proteins and we added the peptide to them via a linker. So the CBD, the cell wall binding domain, is the hand. It will bind to the peptidoglycan. The Pacman symbol is the EAD, that's the scissor that cleaves the peptidoglycan. And this peptide has properties that destabilizes locally and transiently the outer membrane. So you have to imagine it's a kind of drilling device that pushes or that pulls the phage lysin that is fused to it through the outer membrane and brings the lysin in the periplasm where it can bind and cleave the peptidoglycan. And in case of gram-negatives, the peptidoglycan is very thin, just 1 to 3 layers. So you just need a few cuts and the cells will lyse. And this is what happened in a successful way. So a bit more detail you see on the right. So which peptides did we use? And for that, you have to understand how the outer membrane is stabilized. There are 2 major forces stabilizing the outer membrane. First of all, you have the ionic interactions between the calcium and magnesium ions and the negatively charged phosphate groups. That are the strongest interactions. And secondly, you have the hydrophobic interactions by the parallel assembly of this lipid A fatty acids. Okay, so that are the 2 major forces stabilizing the outer membrane. What did we do in the example that I will show you today? It's a lead lysin, and we call them an RT lysin. That's a trademark for this kind of molecule. So we use the peptide SMAP29, and it's an amphipathic helix. So here you see the helical projection, and you see that all the positively charged amino acids indicated in blue are clustered at one side of the helix, and all the hydrophobic amino acids in yellow and green are clustered at the other side. So you have a protruding peptide which is positive at one side and hydrophobic at the other side. And the positive side will interfere with the ionic interactions, and the hydrophobic side will interfere with the hydrophobic interactions. So that's how this peptide can act as a drilling device. So we took a peptide from the innate immunity of sheep, we fused it to a phage lysin of a Pseudomonas phage, and then we ended up with RT lysin 175. This technology is now commercialized by Lisando, and as a disclaimer, I should say that I'm patent holder on this. I'm inventor on this patent of this technology. Okay. So to convince you that it works, I show you this movie. So what happens? We have Pseudomonas aeruginosa cells. We add arteridin 175, and this is what happens. You see that the cells lyse one for one, and this cell is a very interesting one because you see that the lysine, the engineered lysine, punctures a hole, and then the cell eventually lyses. So a few slides before, I've shown you the image with the gram-positive cell, and by engineering, we reach now the same situation for gram-negatives as well. Okay. Another movie I have is with Acinetobacter baumannii, and I show you this movie because the previous one was very hard to record. Basically, what happens when we brought together the cells and the lysins, all the cells were already lysed when we had mounted the sample on the microscope. We really needed luck to find the perfect conditions. Here, the cells are immobilized in the microfluidic flow cell, and we can see in real time what happens if we add arteridin 175. So you see that the timestamp is at -10, and from 0, you will see that the flow of the arteridin will start and will make contact with the cells. And exactly at this time point, you see that the cells lyse immediately, and you see the cell debris and the DNA and the lipids in the stream. So phage lysins and their engineered derivatives are one of the fastest acting antibiotics that exist. Okay. In another movie, I want to show you that these artelysins can be very specific. Here you see a Pseudomonas aeruginosa cell, the rods, and the spherical cells are the staphylococci, the benign flora on your skin. And of course, you only want to kill the pathogen, but not the benign flora. And this is what happens if you add the artelydin. Only the rods are lysed. This one takes a little bit longer, but also lyses, and all the coccal cells remain intact. So you can engineer the lysins that they really work against the pathogen, but not against the other bacteria. And you can do that because the peptide glycan is very diverse, and you can play with this CBD and this EAD specificity, and also with these peptides to modulate and customize the activity. All right. So we have published a bunch of papers about that, and just to summarize it, I can tell you that artelysins work very rapidly. They have a highly bactericidal effect. We have observed up to 8 log reduction when using cells in the stationary growth phase. We found no cross-resistance with at least 12 resistant mechanisms, including colistin resistance. We have tried to trigger resistant development by serial exposure to sub-inhibitory doses, and this always gives resistance for standard of care antibiotics, but not for the engineered lysins, so it's not easy to develop resistance in vitro. And another interesting feature is that they also kill persisters. You know that many antibiotics need an active metabolism in order to kill the cells. That's why they do not work on persisters, which have a dormant metabolism. But in the case of phage lysins, they are active molecules themselves. They are desisters, and they don't need an active metabolism. So they also kill persisters, which are abundant in chronic infections. So that are the nice features of lysins. Okay. I will return to the artelysins, but I first move to the next strategy, and that are lysins with intrinsic antibacterial activity. So it was a surprise, but there are an increasing number of lysins that have an apparent intrinsic antibacterial activity. What I mean with that is that if you add them to the cells, they have a way to permeate through the outer membrane. It's not so efficient, but they get through and they can kill the cells. And the majority of these lysins with this activity, they have a C-terminal amphipathic helix. So amphipathic, they have these positive charges, they have hydrophobic residues, and this helps them to get through the outer membrane. It's a bit, the biological reason for that is likely that when they come from the inside, at the end of the replication cycle of a phage, they also have to destabilize the outer membrane, and likely that's the function of that domain. Another endolysin with this activity that has been reported and has been crystallized has a cationic N-terminus. It has a protruding peptide that interferes with the outer membrane and has intrinsic activity. So while these lysins are really large and cannot pass through the porins, thanks to some feature, they find a way to get through the outer membrane as well. We have recently found a similar lysin, Lys MK34. It's such a lysin with intrinsic antibacterial activity. You don't need engineering, you don't need pressure or outer membrane permeabilizers, but when you add it to the cells, you see a time-dependent effect in the black bars between 1 and 3 log when you add 1 micromolar of this endolysin. But at the same time, we also engineered this lysin with a peptide like we did for the artelysins. And if you combine such a peptide with a lysin with intrinsic antibacterial activity, you can see that you further boost this natural effect and that you get a much faster killing. In the first 30 minutes, you have already reached the detection limit. So you can further enhance this natural property of such lysins. It's not only faster, not only the killing rate is increased, you can also improve the activity against stationary growth phase cells. And the unengineered variant kills it up to 1 log, which is not a high value. But when you engineer it with a peptide at the white bars, you see you get a higher effect. So by combining engineering with lysins that have naturally an intrinsic permeating activity, you combine the best of 2 worlds. Okay. The last strategy that has been followed by us and by many others in literature is using engineering and using a targeted uptake via outer membrane proteins. So what's the strategy? It's based on bacteriocins like pesticin and colicin and apiocin, and the domains that are responsible for the uptake via these outer membrane proteins are used and fused to a lysin, a phage lysin, to get the lysin through the outer membrane to reach the peptide glycan in the periplasm. You can categorize them as lysosins. Our own contribution is enolysins. There we didn't use binding domains from bacteriocins, but we used the binding domain of a receptor binding protein of a phage. And that's illustrated on the next slide. What did we do? It was in collaboration with Professor Lene Bronstedt and a PhD student, Atina Sampara, our postdoc, and it was an initiative from their lab. But they took the PB5, which is the tail tip protein of that phage, that binds to the outer membrane protein, the FUA-A. And if you fuse this protein with an endolysin, we found that this fusion protein also can kill the cells. The activity was not very large. We just had about 1 log killing in 30 minutes, which is not very high, but it worked. So by just bringing the protein in close proximity of the outer membrane, the lysin could find a way to permeate the outer membrane and to kill the cells. The mechanism is not yet understood, but it's still ongoing work. But here you see some images, how it's mainly at the poles that the cells are affected and apparently lead to killing of the cells. I will return later on again to this concept of enolysins. So, second take-home message. Phage lysins are a broad class of antibiotics. And why is that? First of all, phages are the most abundant biological entity on our globe, and there are 2 lysins per phage. So also the lysins are abundant. There is an enormous natural diversity available of lysins. Initially, they were only considered for gram-positives, but there are now an increasing number of strategies available to make them work against gram-negatives as well. And the third feature is, in my eyes, the most unique feature and differentiates them from any other class of antibiotics. They have a modular composition, and we can apply protein engineering to create a practically infinite collection of engineered ligands so that we can make them custom-made, we can customize them to any potential bacterium under any potential condition. And I often make the comparison what happened with the traditional antibiotics. So we had the golden era of antibiotics in the 50s and the 60s. With the Waxman platform, a lot of new antibiotic classes were discovered. But then in the 70s and also in the 80s, it were the chemical engineers that made new generations of antibiotics. So they use chemical engineering, and you got the second generation of cephalosporins, and the third and the fourth, also for the quinolones. They used engineering to make new and improved derivatives. We do the same with protein engineering. We engineer the natural reservoir and we improve their antibacterial properties. And you can do that for any bacterium. So it's really a broad class of antibiotics. So that's what we call the modularity principle. You have different building blocks, the EAD, the CBD, a peptide, and linkers, and you can make a lot of combinations, a lot of different architectures, zillions of combinations, because the architectures can differ, but also the building blocks can differ. However, there was no method to make so many variants, and that's why we have introduced lately the versatile method. The versatile method you can compare with molecular legal. So first step is a versatile cloning step, and you make a box of building blocks. So all the CBDs, all the EBDs, EADs, the peptides, the linkers have to be made compatible with the versatile method. And then you use versatile shuffling, and you can click together all these building blocks. And then you get a collection of engineered ligands that you can test. Okay. So we start with versatile cloning. You have the coding sequence of a certain CBD. We fuse it to position tags that tell the coding sequence in which position it will have to be located. So we do that for different building blocks. We add the position tags, we call them tiles. And then in the second step, versatile shuffling, you link them all together. You assemble them together in the same sequence. So for example, position 1 can be a peptide, position 2 a linker, here a cell wall binding domain, and position 4 an enzymatically active domain. Okay. We clone them in the vector so that we have entry vectors we can use later on. Then we go to the versatile shuffling step. You pick the tiles you want to use. You mix them for position 1. You do the same for position 2, 3, and 4. You do the assembly reaction mixture. You add it together with the destination vector. You do a transformation, and on the next day, you have a plate, and every colony on the plate will have a different composition. So that is what we call combinatorial assembly. So it depends on the number of tiles you include in your reaction mixture, but you will have thousands to even millions of variants with different tiles, different peptides, different linkers, CBD, EADs, and so on. Okay. Then you have a lot of variants, you have a lot of phage license, but then the trick is how can you now find the best phage license? Because most of them will not be functional. You have to look for the needle in the haystack. And for that, we use a microtiter-based screening assay based on growth inhibition. So you see the optical density in the y-axis. So we add the engineered ligand to cells, and only those cells that do not grow, like for example, when you add ligand C8, they will keep a low optical density. The growth has been inhibited. So we screen the variants in this way to find the needle in the haystack, to find the hit molecules, the hit ligands that have a good activity. In yellow, you see some intermediate effects as well. Okay, so we use this platform to engineer a specific phage ligand, which is highly active against Acinetobacter baumannii under human serum conditions, because human serum is a pretty aggressive condition for enzymes. We started with the versatile platform and the versatile technology. We first made the tile repository with all the building blocks with versatile cloning. And then we applied the shuffling technique to make a library with a lot of variants. Next step is the screening. And then you look to the results and you try to find design rules. Which building blocks contribute to a good license? And this knowledge you can now reuse and you can make a new library. You just return to your tile repository, you do a new assembly, you make a new collection of licenses and you screen them again. And you can repeat this as many times as needed. All right, I will drink a little bit. So we made the tile repository with a lot of peptides, CBDs, EADs, and linkers, and we can make all possible designs with that. More than 12 million combinations can be made. And in the first study, we combined 38 peptides, 2 linkers, 6 CBDs, and 21 EADs, and we could make almost 10,000 different phage licenses. So that was a starting point. We used our screening assay, which screened 4 plates, or roughly 389 variants, and we found that 7 of them were active, 2%, and that's a good percentage. 1 was active against 4 strains, but the majority was only active against a subset of the strains. When we looked to the 7 variants, we tried to find design rules, and we started with 38 peptides, but there were mainly 4 peptides that were conserved in the hits. And also at the level of CBDs, we started with 6 CBDs, but there were 2 that mostly contributed to active ligands. So we found 2 design rules that we applied in the next library. Instead of using 38 peptides, we only used 4. We kept the linkers and reduced the CBDs from 6 to 2, and we kept 21 EADs. So we made a new but smaller and a smarter library. We screened it again, and now we had 40 active variants, so a hit rate of 21. And we also had more strain and more hits that were active against all the strains of the test panel. So you see that implementation of these design rules leads to a higher hit rate. Okay. Next step was that then we screened the 24 best ones in human serum to find those that were most active under this stringent condition. And in the end, we had 5 active variants, and 1 of them was active against all strains. So that was our lead molecule. Okay, this lead molecule was performing well in a broader panel of multi-drug resistant strains. It was very specific for Acinetobacter baumannii and not on other gram-negatives or gram-positives. And also it had a very good bactericidal activity. All right. You see, if you look to the movie, that the mode of action of this one is completely different. Instead of inducing complete cell lysis, you see only some local cell wall bulging and a change of the phase control. So we are not yet sure about the mode of action. It works, but it's a different principle than before. In another study, we used a versatile platform to analyze different configurations. Instead of the OMP, the peptide, the linker, the CBD, and the EID, we also made other combinations of building blocks, and we wanted to know which combination is the best to find hits. And this time we focused on Klebsiella pneumoniae, and we made a lot of ligands, and we screened roughly 960 different ligands. And you see that this configuration has a high, important, and drastic effect. Only configuration 1 and 4 had a lot of hits. Those which are on top above this red line are the active variants, and it's mainly configuration 1 with a single OMP, a single peptide in the beginning, and configuration 4 with the peptide in the end, which was highly active. So that's another way how you can use a versatile technique. Also for the inner license, this other concept, besides the arter license, we have used a versatile platform to make different configurations of phage receptor binding proteins and endolyzins with different linkers. We have made a lot of variants. We screened them with 3 different assays to end up with the final variant. And this one had an activity up to 3.3 log killing, while in the beginning we had just 1 log. So you see that this versatile technique can be used to get improved lysin variants that have a better activity. All right, that brings me to my conclusions. So the first shuffling method is this very generic method that can be used to do engineering of the phage lysins and to make variants that have improved antibacterial properties. And it's very simple because you can do an iteration. You do a single cycle, you learn something, you get design rules, and then you can go back to your repository of building blocks, make a new library, and in a cyclic way, you can improve your activity. So we make a bit the classic hit to lead development of small molecule drugs, but then translated to the engineering of phage lysins. This is a classical process in the pharmaceutical industry where the chemists make a library of small molecules, then the biologists design an assay for the goal of that drug, and they do a high throughput screening. They get the output, and then the informaticians will do quantitative structure-activity relationships. They look to which atoms, which functional groups are important in my drug. They give this information to the chemist, and they make a new library. And this is the hit to lead development that precedes the preclinical stage in the pharmaceutical industry. It's exactly this principle where we have made now a functional equivalent for phage lysins, because phage lysins are now in the clinical phase 3 phase for Staphylococcus aureus. It's my true belief that lysins can be developed against any potential bacterium. All right, I would like to thank you for your attention, and also our group. This is the latest picture of a virtual meeting in a corona-proof way. I also thank the many co-workers and collaborators. The names are too extensive in number because I showed you a lot of data, but the major people are all affiliated with Ghent University, KU Leuven, and also the University of Copenhagen in Denmark. Thank you very much for your attention, and I'm happy to take all your questions. Thank you.
Thanks, Yves. Congratulations for this wonderful presentation. Do we have any questions from our panelists?
Yeah, Yves, just a wonderful talk. I mean, unbelievable. You know, the science and the engineering, just incredible. Absolutely loved it. I wanted to ask you some questions. Right away, I was thinking about pharmacodynamics, pharmacokinetics, and also obviously immune response. And so, especially with the endocarditis and the bloodstream infection, Staph aureus license that were utilized, those clinical license, has anything come out in terms of information regarding the immune response to these particular proteins?
Yeah, that's a very good question. There is an immune response. So it's a protein, so antibodies will be raised. We see that in animals. It has also been reported in humans in a clinical study. But apparently, and that's what we know from the animal studies, even if a mouse or a rabbit has been pre-immunized for the endolysin, the lysin, if you infect the rabbit again and you try to treat it with the phage lysin, it still works. So that there are circulating antibodies, but the neutralization rate is slower than the action rate of the lysin. That, at least the commonly accepted hypothesis, is not yet fully demonstrated. But what we know is that there are antibodies, but even if they are there, it still works. The second immune response you have to take into account is that you lyse the cells and that you create cell wall debris. You create cell wall remnants, and that can trigger the innate immunity, and you want to avoid that there is a kind of overreaction on that. And for there, the dosing will be important. It's important that you do not apply, and that's what we know from animal studies, that you do not apply a too large dose or a continuous dosing, because then you will fragmentize the cell wall too much, and then you get an overreaction. So you just have to apply this amount that you kill the cell and not more than that.
And if I could, just one more question. You used human serum with your Acinetobacter. Now, it's my understanding that bacteria grown in human serum actually take up some of the fatty acids. So this is more of a comment. And so my suspicion is they're not quite the same as they would be in laboratory media. Is there I can see why you picked human serum, but have you also tested this, for instance, in laboratory media?
No, the lysins I've shown you today are not yet tested in animals.
No, not in animals, but just in standard laboratory media, as opposed to human serum.
Yeah. So the engineering study I've shown you, the first rounds were in standard laboratory media, and in the final round we went to human serum, because from preceding information we know that there's always a lower number of lysins active in serum compared to in standard media.
I wonder if that reason is due to the alterations in the fatty acid content of the cells.
Yeah, that's something we didn't consider yet. Yeah, we didn't study the effect of this fatty acid composition. Good suggestion.
Yeah, they lose their branch chain fatty acids, is my suspicion with Acinetobacter.
Yeah.
Okay, we have a couple of questions here in the panel. So Jitender Mehta, he's asking, like, as we know, bacteria are rapidly evolving and have several adaptive resistance mechanisms. What do you think about bacterial resistance against lysins? If resistance is develops, any alternative approach solution can be looked?
Yeah, that's a very relevant question, I think, for everyone who is developing new antibiotics. You want to know what's the situation, how is resistance developed. So with that perspective, we did different trials in the traditional way that you expose the cells to, in a serial way, to sub-inhibitory doses of the lysin. And you do it in a serial way, and we use control antibiotics, where with the control antibiotics like ciprofloxacin, for example, you see quickly going up the MIC value, the inhibitory doses. So there are mutations accumulating in the cells, and you see that the MIC dose goes up. Well, this never happened for the lysins. So it's not so easy to provoke resistant development inside the cell by just point mutation. So that's a good benefit. Does that mean that there will be no resistance at all against phage lysins when they would enter the clinic? I don't think so, because the resistome is very large, and I can imagine there are peptide glycan modification genes and enzymes available in the broad resistome, and that can be transferred by horizontal transfer and can emerge possibly as a resistant mechanism. But overall, it looks more promising than most standard of care antibiotics, because this vertical resistance development by accumulation of point mutations doesn't seem to happen easily for lysins.
All right, we have a second question. So Noel Gahamanji, I guess that's the way you pronounce it, is asking that you target Acinetobacter. What about all the pathogens considered as priority for drug development by the WHO? You show their list, but you did not mention how many of them you tested.
Yeah.
Many were gram-negative.
Yeah, I showed you data for Acinetobacter today, but we have published other papers about Pseudomonas aeruginosa. There's also a paper about colistin-resistant E. coli. I've also shown data against Klebsiella pneumoniae, so that are already at least 4 pathogens, 4 species. It's my belief that practically or almost against any pathogen, you can find and engineer a lysin. That's what I try to stress. It's a broad class of antibacterials. It's not just a single molecule. But I think in the coming decades, it will be demonstrated how broad it can really go. But it's my belief that by engineering lysins and by taking lysins from the right sources to start with, that you can create the lysin targeting any pathogen out of interest, not only for human disease, also for veterinary diseases, but also in food conservation, spoilage, crop protection. It can be very broad. Everywhere where a bacterium is problematic, there may be possibly a solution offered by lysins.
Okay, I'm afraid we're running out of time for more questions that you can respond in person. If you don't mind, you have 3 more waiting in the chat. So if it's okay for you, Yves, we can proceed with the next presentation and you can reply them by typing. Thank you again.
Thank you very much, and also for the very interesting questions.
Thank you. And now on, we're going to move with our next talk, which and its next speaker. She is Sara Maria Soto. As a brief introduction about herself, I'm going to say that Sara obtained a PhD in biology from the University of Oviedo in Spain. After a short fellowship as a postdoc in Germany, she obtained a 3-year postdoctoral grant from the Spanish Ministry of Health in Barcelona. And in 2005, she obtained one of the most prestigious grants in Spain, named Miguel Servet, after the great scientist. And that allowed her to establish herself as an independent researcher. She's currently an associate research professor at the Barcelona Institute of Global Health and associate professor at the University of Barcelona too, where she works in the area of viral and bacterial infections, specifically in the subject of antimicrobial resistance and new antibiotics. She's also a member of the the Barcelona Institute of Global Health Gender Commission, director to ensure equity among scientists, which I think is a really important topic nowadays. And she's been coordinator of a Europe 2020 project, No More Film, to find new antibiotics from microalgae. And today she's going to talk about a gold 3 species having antibacterial activity. So let's say welcome to Sara, and I'm waiting for you.
Thank you, Marta, for the nice presentation, and thank you to the organizer to invite me to give this talk about the work that we are doing in our laboratory. I would like to present this drug discovery project that we have in the lab, that is the new gold 3 complexes against multi-drug resistant bacteria. I would like to present a brief introduction about the antimicrobial resistance problem that, as you know, is a worldwide problem that has been considered the top 3 threats to global health by the World Health Organization. It is estimated that about 700,000 persons die annually by infection caused by multi-resistant bacteria. No measure is taken in the next years. It is estimated that in 2050, more than 10 million will die from antimicrobial resistant related infections. 2 million more than this. 2 million more than the deaths by cancer diseases. But in addition to the problem of the emergence of bacteria, of the resistant bacteria, there is another problem, that is the decrease of investment for the pharma companies in the development of antibiotics. This is for several reasons. One of them is the treatment with antibiotics is very short, normally between 3 to 2 weeks, and the revenues for this treatment is not so high that the treatment for chronic infections. For this reason, we need urgently new antibiotics with new mechanisms of action. As commented previously, Yves, the development of new antibiotics has the boom with the 30s and the 60s, more or less 70s, and after that, after the development of a new antibiotic that they use in the treatment, in the clinical treatment, after sooner or later we have bacterial resistance to these antibiotics. It's very clear in the case of methicillin. Methicillin went to the market in the 60s, and in the 62 there was the first Staphylococcus aureus methicillin resistance. And that's what happened with all the antibiotics that we have in the market, including the last, like linezolid and daptomycin as the last one. 2 weeks ago, the World Health Organization has published a report about the antibacterial products that are in clinical and preclinical development. In the case of clinical development, we have 60% of the products are antibiotics and 40% are non-traditional products. Among the non-traditionals, there are antibodies, micromodulation, bacteriophages, as Yves mentioned, immunomodulating agents. And from that, 43 are against priority pathogens, 12 for Mycobacterium tuberculosis, and 13 for Clostridium difficile. In the case of preclinical pipeline, there are more products. In this case, there are 52% of the products that are specific for one pathogen, and the 48 are not specific, they are broad-spread. And for the number of products by pathogen, you can see that the more predominant is for Enterobacteria, followed by Pseudomonas aeruginosa, Staphylococcus aureus, and Acinetobacter baumannii. In Acinetobacter, they are in the preclinical pipeline about 58 products. What happened with gall in our case? How is gall as antibiotic? The first studies were carried out by Robert Koch at the end of the 19th century, investigating the activity of potassium isonarate against Mycobacterium tuberculosis. In the present, there are a relatively large number of gall-1 complexes that have been synthesized and studied. In contrast, although gall-3 complexes have been intensively investigated as anti-tumor agents, there are very few studies related to their antimicrobial activity. Some examples, there are the bicyclomethylate gall complex 3 against E. coli, Bacillus subtilis, Pseudomonas aeruginosa, and Candida albicans, or some ditocorbamate derived from amino acids that are active against Streptococcus pneumoniae. What is our product, our molecule? The initial product is a cationic mixed cyclomethylated gall-3 ditocorbamate complex that have this structure. It's a general structure. For this initial complex, the chemical part, the research group of this project, that is the University of Almeria, has elaborated 16 products with variation in the ditocorbamate ligand. These products have a minimal inhibitory concentration value for Acinetobacter baumannii between 4 and 52 micrograms per milliliter. Well, we had from this product, we select one that is the UL267 that is considered for us is our hit compound that we are developing now. This is we test this compounds against a lot of clinical isolates, clinical strains that we have in our laboratory for a lot of hospitals in Catalonia. We test against Pseudomonas aeruginosa, multidrug-resistant bacteria. Also, we have the ATCC strain as control, Acinetobacter baumannii, including a pan-resistant strain, E. coli with a multidrug phenotype, Staphylococcus aureus, including methicillin resistant and methicillin susceptible strains, Stenotrophomonas maltophilia, and also Haemophilus influenza. And you can see here, there are the MIC that we found with some of these variant molecules. This is I remarked here the UL267. As you can see, they have some mixed for Pseudomonas aeruginosa of 8 micrograms per milliliter. In Acinetobacter baumannii, we find an MIC very, very low of 0.06 in the strain that has a mutation in the lipopolysaccharide. And in other strains, also multi-resistant, they have an MIC of 8. In the case of Haemophilus influenza, the MIC range from 0.5 to 1. In the case of E. coli, 4 to 8. In Staphylococcus aureus, in methicillin-resistant Staphylococcus aureus, you can see it's also very low, from 0.06 to 0.5. And in the case of Stenotrophomonas, the MIC is about 1 to 2. Therefore, our molecules is active against the high priority, the critical priority from the CDC, Acinetobacter, Pseudomonas, Enterobacteriaceae, also against Staphylococcus aureus, the high priority considered as a high priority, and also with medium priority, Haemophilus influenza. I had to say that we don't know if this also active against other species that are in this high priority group, because we have not tested yet. We also do the cytotoxicity in human cell lines. In this case, in URCAP is 6.1. You can see this is the DMSO, that is the product that we use for solubilize the molecule. And this is our molecule. This is the 267. It has an IC50 of 4 milligrams per liter in this case. But also, another test that we do it is the hemolysis ability to decompose, because it can break the erythrocytes. In this case, we use the erythrocytes from healthy donor, normally people from the lab that they are very, very collaborators in this sense. We have a treatment of 4 hours with our molecule. We finally centrifuge the erythrocytes, and we reach the supernatant absorbance of 550 nanometers. And you can see here, this is the baker, and this is our compound. And we find at a concentration of 32 milligrams liters, only the 10% of hemolytic activity. It's not so toxic, but we had also to know how many times our compound kill the bacteria. And we perform for this reason the time kill curve. This is in the case of Acinetobacter baumannii, pump-resistant strain. We use several concentrations of our molecule, the MIC, the two-fold MIC, and we can see that when we use the two-fold MIC, the time that the molecule needs to kill all the bacteria is about 4 hours. It's very fast in this sense. The problem is that if you have an antibiotic, you market antibiotic, and in 10 years, you find bacteria resistant to the antibiotic, we would like to perform to know if our antibiotics, our molecule, got pre-complex, is able to generate a resistance in the bacterial strains. We perform these experiments with 1 gram positive and 1 gram negative, in this case, Staphylococcus aureus and Acinetobacter baumannii. In the case of the Staphylococcus aureus, we perform also the single-step mutagenesis, and we don't find any mutant. And after that, we submitted the susceptible strain to 30 days to different concentrations. And we used also the control of daptomycin. And this was very surprising because our molecule doesn't change the MIC. Sometimes one fold the MIC, but it's possible for a pipette error. But you can see as the daptomycin, after the 30 days, they have increased the MIC 10 folds more than the first one. It's only they have developed rapidly this resistance. In the case of Acinetobacter baumannii, it was the same. After 30 days submitted to different molecular concentrations, we use the control, the colistin, and you can see that. You can see as our molecule hasn't generated resistance after 30 days, and the colistin increased rapidly to 11-fold more the MIC than in the first day. For this reason, this is a good thing for us. It don't generate the resistance, but also it's a bad thing for us because we don't have a mutant to compare to the wild type to know where the molecule is acting in the bacteria. For the reason to determine the mechanism of action, we began discarding well-known mechanisms of action. The first one was the gyrase. As you know, quinolones, the gyrase A is the first target for the quinolones in gram-negative bacteria. To know if our molecule acts in the gyrase A, there are ECA, that is a commercial kit. It's based in the activity of the DNA gyrase, that you have a release DNA, the activity of this enzyme transform this released DNA in supercoiled DNA. The procedure is very easy. You mix the reagents for the kit with this released plasmid, and you include edit the compound that you have to test in different concentrations. Do preheating and edit the gyrase. And after 3 minutes of incubation, just stop the reaction and you see the results in a Gaussian. This is a possible result. Is you find all the open circular release topoisomers here in the up part of the gel. is that the DNA erase has no acts. They don't transfer the relation DNA to supercoiled DNA. But if you find here in the bottom part of the gel, the bond is that all they are supercoiled topoisomers, and the DNA erase had carry out his action. In our case, this is our case. This is the ciprofloxacin, and this is our molecule, the UL267. And in this case, you can see that most part of the relays DNA has transformed into supercoiled DNA. Here you can see the graph, and you can see this is the activity of the ciprofloxacin, that after our concentration of 2, they are more or less 25% of the inhibition of the activity. And in the case of UL267, that is with the square, there is no modification of this activity. The other mechanism is also similar to Lloret's activities, the topoisomerase 4 activity. The topoisomerase 4 transforms the kinetoplast DNA in the catenated key DNA. And you can see the procedure is similar to the Lloret's. It's also a commercial kit, but with the difference that you use kinetoplast and the topoisomerase as enzyme. And you can also see the results in the agarose gels. If all the DNA that you use is in the wells, it refers that the topoisomerase does not do the action. But if you see the band at the bottom of the gel, is that all the kinetoplast has been transformed in the catenated key DNA, and the topoisomerase has acted. But in our case, it's the same. They have no effect in the topoisomerase. You can see here all the catenated DNA in the bottom of the gel, and you can see here also in the graphic that the UL237 maintain the 100% of topoisomerase decatenation. Okay, our molecule has now the mechanism similar to the kinolans, but other that we have we test is the efflux pumps. You know that the efflux pump go out the antibiotic from the for the cell to the exterior part. In this case, we make the experiment using phenylalanine, arginine, beta-naphthylamide, and rocepin. And you can see in the table that the MIC for Pseudomonas aeruginosa, E. coli, strain Trophomonas, Haemophilus influenza doesn't change when we use the inhibition, the efflux pump inhibitors, that's the PAN and the rocepin. Therefore, we discard also the efflux pump. Finally, we show the cell, what happened with the cell, and we test them by transmission electron microscopy. And we were very surprised because this is the case of Pseudomonas aeruginosa. You can see here the string without treatment. It's very straightforward. And you can see here with the UL267 treated only 2 hours. You can see here a lot of cells empty, all the cytoplasmatic continues here, and here is losing the cytoplasm, and in more you can see here the normal cell with the membrane, intact membrane, and you can see it has some part of the membrane are totally broken. But we don't know if the molecule is entered to the cell and after doing the action into the cell go out, or they broke directly the membrane. As one of the trends that they have in the clinical setting is to do combinatorial treatments. In the case of multi-resistant bacteria, we also test the ability to have synergy effects with our molecule and the antibiotics used in clinical treatments. We test ciprofloxacin, quinolones, beta-lactamics, and finally we obtain synergy with colistin and some aminoglycosides as amikacin and gentamicin. In the case of colistin, we use, in this case, because we had the span resistant Acinetobacter baumannii strain that is colistin resistant too. And as you can see, for MiCO267 in the case of the Gold-3 complex and MiCO colistin of 32, when they are used in combination, they decrease this MiCs to 0.25 to the gold complexes and 1 for colistin. The same happened in the other strain that is also resistant to colistin. They reduced to 8 to 0.5 in the case of the Gold-3 complex, and for 16 to 2 in the case of colistin, having a FIC very, very low, the 0.1 being considered synergistic with 0.5. Also happened in these that were no colistin resistant, but they decrease also a lot the concentration of those molecules. This happened in microscopy. You can see this is Acinetobacter baumannii, that is intact without treatment. When we treat with the UL267 with the gold complex, you can see that the membrane is more diffuse. You have here losing a part of the membrane. You can see here that some of the cell that I don't know if this one or another one is losing the cytoplasm. The corpuscular here that is very, very cheap. They are here that lose the cytoplasm. The colistin alone produce these vesicles around the cells. You can see in all the cells. And when they combine the Gold-3 complex of the colistin, you can see have lose the material, the plasmic material, and it made this empty part inside the cell and also the vesicles. And it's very nice, this effect. What happened with the bacteria? How many times they need to kill the bacteria in combination? But as you commented previously, we need to fold the MIC of the Gold-3 complex to kill the bacterium Acinetobacter baumannii, in this case, in 4 hours. In the case of combination, we need less concentration of both molecules to kill the bacteria. And in this case, with the half MIC of both molecules of gold complexes and colistin, you kill the bacteria in only 2 hours. It's very, very fast. In this sense, we think that the colistin help our molecule to penetrate in the cell. Well, we know that it has antibacterial activity. We know that it has a moderate toxicity in the case of for gram-negative treatment, but the window of toxicity is good for gram-positive bacteria. But what happened in vivo? Do you know that when you are testing some molecules, sometimes the good result that you have obtained in vitro is very different that you obtain in vivo? It's completely different. This is the scheme that is presented from a lot of molecules. Molecules, you go decrease the number until you have the lead. No, sometimes the problem is in vivo, in the behavior of the molecule in vivo. We made the acute toxicity in mice. This is a preliminary proof of concept using CD1 female mice of about 8-10 weeks of age and 30 grams of weight. We give to the inoculate a single dose of 2, 5, and 8 milligrams per kilo of intravenous. And we use 5 mice per group. And we test symptoms of toxicity, weight loss, weight coat, poor motility, or lacrimation. As you can see, with the doses of 2 milligrams per kilo and 5 milligrams per kilo, we don't observe any loss of body. And after 9 days, we have the 100% of survival of the mice. But with the dose of 8 milligrams per kilo, only 40% of the mice survive after 9 days of treatment. We have estimated here between 5 and 8 the acute doses of toxicity. And here, after that, we made the necropsy of the mice, and we also observe and weigh the organs, because you can see there are some effects in the organs by the coefficient between organ weight and body weight. And we test the spleen, the kidney, the lung, and the liver. And you can see there are no significant difference with the control and the mice with the treatment with our core complex. This is not in the spleen, not happening in the kidney, lung, and liver. The weight and the visual recognition is the same in both. We have here, but we would like to know if our molecules is at least effective, active in vivo, no? Because as commented previously, you can have a good results in vitro, but when you go to the mice or to the animal models, you don't obtain any effectivity. You don't are able to kill the bacteria in the body. This is a preliminary study. I would like to remark that we don't know the pharmacokinetics of the molecules. It only take into account the studies on toxicity that we previously commented. And here we used also the CD1 femal mice, and we have the control group of infection that we make an inoculo of bacteria. This case is methicillin-resistant Staphylococcus aureus. And we have the treatment groups. That was one with a single doses of 5 milligrams per kilo. And the other one in green is the multiple doses of the 5 milligrams per kilo each 24 hours. And you can see the control, all the mice died in 2 days. With a single dose, they are surviving at least until the 4th day, until the 5 days, at least the 20%. And finally, with a multiple doses, all the mice die at the 4th day. It's not so impressive results because it's only a preliminary study as is the heat, but we can see that at least our molecule has a positive effect in the treatment of this sepsis by Staphylococcus aureus. Where we are in the, for me, it's the worst phase that we had, that is the heat to lead. We are now with computational chemistry, making some changes in the molecule and to try to solve the problems and to decrease this toxicity and to increase also the antibacterial activity. These molecules are under a European patent in June 2020, and also in a PCT application that now is entered in national phases in different countries in Europe and also in America and Asia. And this is our next steps. Now, as commented, we are here making computational chemistry design because we have may also have toxicity. We saw that the main problem that have the molecules that they inhibit some cytochromes. And this is the main problem of the molecule. And we now are making some changes in the molecule that is very, very easy to modify. And there are a lot of parts of the molecule that you can modify. There are 4 parts that you can modify the molecule. And now they have a list of these designs, and now we are making the synthesis. After the synthesis, this molecule, we had to test the antibacterial activity, and the active one we submitted to ADMETO, another time in order to know they have solved these problems. After that, after all these things, we will perform another proof of concept with in vivo acute toxicity also in vivo efficacy of the new molecules to nominate a lead. And when we nominate a lead, the second part is to make the resynthesis, to establish the pharmacokinetics properties. Another time with the new molecule, we have to determine how they are able to generate resistance in the bacteria, and also many times they need to kill the bacteria against the toxicity and against a best extended mouse model, sepsis infection model with taking into account the pharmacokinetic properties. Finally, the preformulation and the metabolite identification in the mice, to have the lead candidate to enter in preclinical phase. It's a long process that needs a lot of money. This is the problem for academia in this case. No? In conclusion, we had developed an innovative class of antibiotics, of metal antibiotics, that is a new chemical class. They are gold-free. This type of gold-free acid, you can see in the samples, there are some molecules, but with this structure is the only one. This is effective against multi-drug resistant bacteria, including the most critical pathogens, gram-negative and gram-positive. They is enabled to generate resistance in gram-positive and gram-negative bacteria. Also, they are absence of co-resistant to existing antibiotics. And I think we think that is a promising new antibiotic to fight against multi-drug resistant bacteria. Finally, I would like to thank to Carlos Ratia, that is my PhD studying as the hands in the lab. The University of Almería, due to the clinical part of this project, the Instituto de Salud Carlos III for the funds to carry out the project, and the Cerca Gingell for the Catalonia government to give us money to do the patent and to conserve him. Thank you for your attention, and sorry for the informatic problems. Yeah.
Thank you, Sara, for this wonderful presentation. No worries for the informatic problems. I guess we all having that. Just a thing I want to share. Isn't it satisfactory to see bacteria to explode? Like in your pictures, in the previous presentation, oh my gosh, I love those fireworks. And I think Metallica would have enjoyed that too.
Yes. It's seen that you have a gun on the
Yeah, it looks really mesmerizing and relaxing and satisfactory at the same time. I don't know if it's my sadistic side. Anyway, I want to ask first the panelists if we have any questions for Sara.
Yeah, I just got an odd question. Do you know where gold binds in bacteria?
We are not yet sure, but now we are doing some experiments because we think that they bind in the teichoic acid of the bacteria. Okay. Because the thing that in the gram-positive bacteria, the MIC is lower than in the gram-negative, and with the colistin help our molecule to be more active, we think that is this teichoic acid, this membrane that is binding the molecule.
So it's not going to bind to a protein, it's going to bind to something else.
Yes.
Yeah, that's nice.
But it's complicated for us because normally when we have a molecule, we use the wild type and the mutant one to compare. And it's very easy because the change in the mutant, you know, that is due to the molecule. But in this case, it's complicated because we don't have mutant.
Okay, thank you.
Thank you.
Okay,
very nice talk.
Thank you.
Okay, please.
So, yeah, Sara, thanks for the talks. So teichoic acid, as you know, is present in gram-positive bacteria, not in gram-negatives, and it's also not essential. I mean, if you inhibit synthesis of teichoic acid, that becomes essential because there's a corrupted product. But simply deleting the pathway, for example, for biosynthesis of teichoic acid, that is not essential. So I don't think that that's your real target. I mean, I understand that the gold is going to bind, but whether or not you have a target, I mean, probably the most straightforward way to address that is to see if indeed you have resistant mutants. If you don't have resistant mutants, that means that you have more than one target.
Yes, we think that we are in front of more than one target. Now we are today we have received the results of internalization of the molecule. This internalized very quickly, and for the reason I managed two ideas. One is directly to the membrane, but the other one is that they internalize in the cell, and after the action in the cell, they go out and broke the membrane or something like that. We don't know, but I think in the next month we can do another conference and we explain the target of the goal.
Hey, Sarah, thank you so much for the talk. I greatly appreciated it. It was very focused, in terms of trying to determine the activity. On the tail end of your kill curves, where you had the control as the antibiotic, I forget the drugs that you used, but you also had your gold conjugate. And what I saw is that you had colonies actually surviving at the end of those treatments. And I was curious if you ever sequenced the genome of those particular survivors to determine whether or not they actually had mutations, I would presume in intrinsic drug resistance mechanisms of some kind.
We are doing that. After we had no clear mutant, we are making transcriptomics of the wild type and the strain after the 30 days of submission of the molecule in order to see if they are something affected.
I was thinking just from a genome perspective, a comparative genomic as opposed to a transcriptomic to identify mutations. So
yeah.
Thank you, Sarah.
No, we have also the possibility. Thank you.
Okay, Sarah, we have here a question in the panel from Dustin Maidanuk. He's asking, he's from Cardona Lab at the University of Manitoba, Canada. So he's asking if your gold-3 compounds are as prodrugs releasing the gold once they enter the bacterial cell. I think what he refers to is like, yeah.
Yes, this could be that they enter and they go outside, but we have to confirm. I don't hear you, Marta.
Sorry, do we have more questions? Or I have a question actually, because I'm currently, it's time for me. I'm currently working on a project involving gold compounds too. So do you even thought about, or are you trying to test those gold complexes towards cancer activity too?
In this case, we are more focusing in antimicrobial activity, but we are in the first compound we test as anti-tumor also. But as our lines antimicrobial resistance a bit, we had another research group in Asturias, in Spain, that made also anti-tumor, but they now are very focused in another molecules and flavonoids and something like that. And
I mean, since you have the compounds, why not recycling them and trying something else? Yes. Another thing that I was going to suggest you, did you thought about any kind of a drug delivery to improve the penetration of these gold complexes?
Yes, we are working in liposomes, with liposomes in this case. Yes.
Okay, if we have no more questions?
Yep, just one.
Go ahead, Tony.
Sarah,
tell me.
You start with gold-3, but you have a gold-1 oxidation state available. So when you get into your nice little cell or your bacteria, what is the oxidation state of the gold?
They don't change.
You don't know?
No, they don't change. The state doesn't change. We are tested by XPS.
Okay, so your son is gold-3.
Yes, gold-3.
Okay.
It was very surprising because in our first idea, we think that they was to gold-1, but no, it doesn't change.
Okay, because with silver you go from silver 0, silver 1, silver 2, and a couple of other ones are available. I was just curious.
Yeah, that's weird because usually they convert to gold-1 easily, and that's why.
Yeah, you can get gold
nanoparticles. Sometimes they cover to nanoparticles. No, it's not the case.
Yeah,
it was very surprising. It's very stable in the sense.
Switch oxidation states.
Yes.
Okay. Well, I think I'm going to thank you, Sara, again for your talk.
Thank you. It was a pleasure.
So much. And I think we were moving forward with I'm giving back the speak to Tony, and he's going to continue with this session.
Okay, thank you to the two previous speakers. Thank you, Marta. I am now going to take over, and unfortunately, you're going to have to suffer from me for a couple of talks. Our first speaker will be Professor Kim Lewis, who's at the Antimicrobial Discovery Center at Northeastern University in Boston. He's done an awful lot, so I won't go into his biography too much, apart from mentioning that he has managed to found several happy companies, which is always a mark of somebody doing something that works, and not an academic. So, Professor Lewis, I will turn over the speech and the function to yourself, and you have 40 minutes, please.
Thank you, Tony. All right. So how to beat antibiotic resistance. This is, as you can imagine, an aspirational title, and I'll tell you how we're going about that. But first of all, let me start by saying, why is it so hard to discover new antibiotics? A couple of reasons we already heard at this conference. There was a terrific talk by Helen Skórska about the penetration barrier in gram-negative bacteria. This one, I don't think we touched upon yet. There's an enormous background of toxic and known compounds. You see, it is very easy to discover antimicrobials. A natural product library will have a hit rate of 30–40%. Antimicrobial activity from a library of random extracts. A chemical library will have, 5 to 10% hit rate against gram-positive bacteria. So that's very easy. What is very difficult is to find a novel target-specific compound from which you can then hope to realistically develop drug candidates and drugs. Another important barrier is actinomycetes, the traditional source of antibiotics, have been overmined. I don't think that group has any useful compounds left against gram-negative bacteria, and I have a reasoning to prove that, but not the time to explain it at the moment. There is then a very high frequency of resistance to compounds hitting single targets. So as a result, most of our compounds in the clinic currently target 3 targets. So the universe of approximately 500 available essential targets has collapsed to 3 or so. So cell wall, the ribosome, and gyrase topoisomerase. So the result of that is a last new class of antibiotics against gram-negative bacteria, the fluoroquinolones, was discovered in the 1960s. Now, ignoring these obstacles, let us ask ourselves an obvious question: what is it that we would like to have ideal, ignoring reality? Well, here's my short wish list. It would be nice to have antibiotics to kill tolerant persister cells, to cure chronic infections. Persister cells are not only responsible for recalcitrance of chronic infections, the treatment, but also are causal in many cases to the rise of classical resistance. So Natalie Balaban has nicely shown the link between the two. It would be nice to have antibiotics that avoid resistance development. That would solve our antimicrobial resistance crisis. And it would be nice to have antibiotics, of course, acting against gram-negative bacteria. That seemed to be a particularly daunting task for us. So I'll tell you how our team was addressing this wish list. So here's a textbook representation of resistance mechanisms. There's a whole bunch of them. Probably all logical possibilities have been realized in nature. And these are very well understood. And these are also the spread of these resistance mechanisms is the cause of the antimicrobial resistance crisis we currently find ourselves in. What is not so well understood is the alternate phenomenon of drug tolerance, where you add an antibiotic to a population of bacteria, and if there's a resistant cell, it continues to grow in the presence of antibiotic. the bulk of the susceptible population collapses, and then you hit upon the small subpopulation of drug-tolerant persister cells. So of course, it would be nice to know why they are tolerant to killing by antibiotics, and what is it that we can do about it. So I'll tell you briefly, I'll make a brief introduction into the basic science of persisters before we go on to treatments. I'll show you an experiment. This is from a paper that just got published from my team in collaboration with Johan Paulsen from Harvard Medical School. And so what we're doing here is we're testing a guess that persisters are low ATP cells. And we're testing this with the help of this ATP sensor. So this is single cell analysis in a massively parallel microfluidic device. We're tracking 900,000 cells simultaneously, but I'll show you of those 900,000, I'll show you only 2 cells. So here is a representative of a regular cell in a stationary population of E. coli. So it's light blue, so it has relatively low ATP. Blue is low, red is high. And here is a cell, a mother cell that's produces progeny at some point, but then now it is very, very low ATP cell. And we're suspecting that this may be a persister, a low ATP cell. And let's see what happens with these 2 cells and their progeny as we add fresh media. And apart from adding fresh media, we also add an antibiotic. Now let's see. Okay. So as you see, the regular cells started growing, acquired ATP, and then this cell and its progeny was lysed by ampicillin. This cell, the low ATP one, is a persister. It slowly starts acquiring ATP, all its progeny lysed. This one survives. And then when ampicillin is washed away, it starts acquiring ATP, it resuscitates and builds a new population. So persisters are indeed low ATP cells. So the question is, what's the mechanism responsible for that low ATP? So here we were thinking also, this is a pretty simple idea that a random fluctuation in expression of energy producing components in the population may lead to low ATP. Here's a test of that hypothesis. So we take Krebs cycle enzymes and do translational fusions to YFP. And first of all, examine these translational fusions by FACS analysis. And what you see that for every Krebs cycle enzyme we examine, there's a very broad distribution of expression of that Krebs cycle enzyme in the population. You have some cells that have extremely low levels of Krebs cycle enzymes, and here's the medium, and this is high. This is a logarithmic scale, right? So now we ask the question, what is the relative susceptibility to killing of these different subpopulations? And what you see, for example, here is succinate dehydrogenase. So these are cells with very low level of expression of succinate dehydrogenase. These are DIM. And the DIM cells, let me reacquire my laser pointer here. And the DIM cells survive much better than the bulk of the population. So this provides you with a mechanism of persister formation, a formation of these low ATP cells. So how is low ATP linked mechanistically and causally to drug tolerance? So what we know about bactericidal antibiotics that hit these major 3 classes of targets that I mentioned to you, what we know about bactericidal antibiotics is that they act not by inhibiting their targets, but by corrupting them. So these are energy or ATP dependent processes, of course. And for example, aminoglycosides kill the cell by interrupting translation. That creates short misfolded peptides, which are toxic. That's what kills the cell. So you have high ATP, bactericidal antibiotics kill very effectively. If you have low ATP in a cell that happened to have a low level of expression of a Krebs cycle enzyme, for example, you have low target activity. it becomes a persister, it's tolerant to antibiotics. So it survives killing by antibiotics. So this simple model is based not only on the experiments I showed you, but on a fairly large body of work from my lab over the years. We think that this is a general mechanism that explains persister formation in bacteria in general and drug tolerance, and also tells us exactly why our antibiotics do not kill persister cells. While it is satisfying to understand why, it doesn't tell us much about what to do about it, apart from perhaps underlying the point that this is probably the toughest problem in antibiotic discovery. How are you going to find an antibiotic that is supposed to corrupt a target without any requirement for ATP? Well, luckily, once you realize these things, there is an old abandoned antibiotic that comes to mind. That is acyl depsipeptide, discovered a long time ago by a team from Eli Lilly, and then a group from Bayer made a more active analog, ADEP4, and also determined its principal mechanism of action, which attracted our attention. So ADEP targets the CLIP protease in bacteria. The CLIP protease normally recognizes misfolded peptides with the aid of ATP-dependent chaperones that feed that peptide into the protease proper for degradation. In the presence of ADEP, the pore of the protease opens and it can accommodate peptides and digest them without the need for ATP. So this is the key, right? Here is a target that you can corrupt without requirement for ATP, because proteolysis per se does not require energy. So, our first test of this was to see whether in the presence of ADEP, you can see proteolysis of mature proteins in a non-growing cell. Now, in Staph aureus, we have shown that the stationary population is essentially equivalent to persisters. It's virtually completely tolerant to killing by conventional target-specific antibiotics. So in this population, ADEP causes a massive degradation of over 400 mature proteins. So ADEP forces the cell, if you will, to self-digest. And here is a test of a simple proposition that ADEP may be an anti-persister compound and perhaps evolved to do exactly that. So here's a control. Staph aureus treated with ciprofloxacin, and here's a population of surviving persisters. Same happens with rifampicin. And with ADEP, you get complete sterilization of this population with the killing of regular and persister cells. Now, we also tested this in a very tough model of an incurable chronic infection. So this is a neutropenic mouse with a knocked out immune system. A massive biofilm of Staph aureus is growing in the thigh. This is not a standard thigh model. This is 24 hours after introduction of the pathogen. And if you allow that biofilm to form, okay, so this is a regular treatment with regular antibiotics, including their combinations. And once you eliminate regular cells, persisters survive, and there's not much you can do about this. Now, in the case of ADEP, with rifampicin, you get complete sterilization, and we need to add rifampicin because ADEP alone, it starts to kill, but then it acquires resistant mutants. That's why the compound was dropped, not developed by the industry. The CLIP protease is not essential. So null mutants arise with high probability and confer complete resistance to ADEP. If you combine it with another antibiotic, the resistance development, then you get complete sterilization. The compound itself is not terrific from a pharmacological perspective. It has high protein binding, poor metabolic stability. So it's a biotech company, Briers, is now developing advanced analogs of ADEP to move into clinical testing. Hopefully this will become a drug against chronic infections. What else I want to tell you is what we learned about persisters from this experience is apart from ADAP that we borrowed, we also found a novel compound, lasomycin. This came out of a screen that we did. It was a selective screen against mycobacteria using a library of uncultured producers. So this compound, lasomycin, hits the ClIP-C chaperone of the unique mycobacterial ClIP protease. And what it does, it forces the ClIP chaperone to digest ATP in a futile cycle. So it depletes ATP apparently to the point of no return, and mycobacteria die. That's how it kills regular cells and persisters. This is another anti-persister compound. Interestingly, these two compounds are attacking the same target. I'm not sure why. It's an interesting puzzle. But what these two examples, and these are only two, right? I don't know of any third compound that is target selective and kills persisters. So far we have these two examples. But what these two examples tell us is that the general approach to kill persisters is to activate hydrolysis. In this case, hydrolysis of protein, in this case, hydrolysis of ATP. But hydrolysis doesn't require energy. That's sort of the brilliant solution to the low ATP persister mode of defense. That's how Mother Nature beats that defense. And there are lots of hydrolases in the cell that are important. There are DNases, RNases, lipases. Of course, they're all tightly regulated. And so dysregulating any one of them will probably give you an anti-persister compound. Okay, so in the remaining time, I'll tell you about, more specifically about two novel compounds that we discovered and are working on, sort of moving them into development. You've already seen this timeline of antibiotic discovery, I think twice during this meeting, so I'm not going to belabor the point. I just want to very briefly mention two important milestones, if you will. So one is here is discovery of daptomycin. This is the last class of antibiotics that has been discovered. So that's in the 80s. And this is the last discovery of a class of antibiotics capable of acting against gram-negative bacteria. This is 1960s. So, and this, of course, is the golden era of discovery, where most compounds that were discovered came from soil actinomycetes. And this seems like a heavily overmined group. And it is the overmining of actinomycetes that caused the end of the golden era and placed us in our current sorry predicament. So thinking about where to get new antibiotics, we decided that we need to go outside of actinomycetes. And for starters, we decided to target uncultured bacteria. Uncultured bacteria is, of course, not any particular taxon. It is just a puzzling phenomenon where 99% or so of environmental bacteria do not readily grow on our petri dishes. So with my colleague Slav Epstein, we decided to develop a method to grow uncultured bacteria. The idea is very simple. You grow them where we know that they will grow, which is their natural environment. So for that, we take a sample from marine sediment or soil, mix it with agar and sandwich between these two semi-permeable membranes and glue the ring. And then this contraption, which is a diffusion chamber, goes back into its natural environment. So now everything diffuses through these membranes, and the bacterial cells inside the chamber are tricked. They think that this is their natural environment, and they produce colonies. So recovery by this method is about 50%. And once colonies form in the diffusion chamber with high probability, they will form colonies on a regular petri dish. We call this domestication. And so these domesticated and cultured bacteria, then of course you can screen them in a standard way for zones of inhibition. In this case, it's a petri dish overlaid with Staph aureus. So we've been collaborating with Novo Biotech Pharmaceuticals, biotech company, have now more than 35 novel compounds, and this is our current lead compound. Current lead compound is teixobactin, produced by this very interesting bacteria, formerly uncultured, Eleftheria tsara, belongs to a new genus of bacteria. It is a beta proteobacterium, so it's gram-negative, right? Not very famous for producing antibiotics. This is an interesting compound. It has terrific activity against the all gram-positive bacteria that we tested, including tuberculosis. We're now also, testing it in animal models of TB. The compound is not active against gram-negative bacteria, with the exception of Haemophilus and Moraxella. And we, of course, the first thing that we usually do when we have a new compound, we raise resistant mutants. First of all, to find out what the frequency of mutations is, and then to find the target by sequencing the genome of resistant mutants. That did not happen for teixobactin. We got no resistant mutants, which usually tells you that you are dealing with a detergent, that you discovered a new version of soap. Which in our practices, the majority of natural products are membrane acting and toxic, not very useful. But this compound had no site toxicity. So that became very interesting. And we did an evolutionary experiment which maximally favors the selection of resistant mutants. So the way this works, let's have a look at ofloxacin, which is our control. You start with a sub-MIC concentration of the compound. You grow a population in liquid media, and then any mutant, even slightly more resistant than the rest of the population, will be able to grow then on a slightly higher concentration. So you up the concentration every day, and in about a month, you get very high levels of resistance. And this is pretty much how all antibiotics in the current clinical practice act in this experiment. With teixobactin, there was no change in resistance. So that was telling us that teixobactin probably does not hit a protein target, because proteins will mutate to diminish binding of antibiotics. And we figured that this would have to be probably a precursor of a polymer such as peptidoglycan. There isn't much else out there really. So we were focused on testing peptidoglycan, precursor, which is lipid 2. And in collaboration with Tanya Schneider from University of Bonn, who's an expert in peptidoglycan synthesis, we determined that indeed teixobactin hits lipid 2 and also a lipid 3 precursor of uraticoic acid. They're very similar. Here's just a closer look at what is happening. Neither lipid 2 nor lipid 3 are mutable. So these are immutable targets, and the moiety that teixobactin binds to is a pyrophosphate sugar, also immutable. You cannot change either pyrophosphate or sugar. These are absolutely conserved in bacteria. If you start changing them, you probably will not have peptidoglycan. Now, recently there was a nice paper from Marcus Weingarth that came out last year, showing the details of teixobactin action using solid state NMR. So this is not real teixobactin. Unfortunately, this is a synthetic analog, but I think it's good enough. So what he found is that once teixobactin binds to its target, it forms this antiparallel beta sheet, a supramolecular structure. So this is a a vast, our common paradigm of one ligand, one target for antibiotics. Here, an antibiotic forms a supramolecular structure which is visible in a regular microscope. And we're now collaborating with Marcus. We have a uniformly labeled native teixobactin, and he finds some additional interesting modes of action of teixobactin, which I'm not going to talk about today, but the paper should come out later this year. So this is the first example of a antibiotic that is free of detectable resistance. I don't think that resistance will develop to this compound, based on its mechanism of action, what we know about this compound. The unfortunate downside of teixobactin is that it only acts against gram-positive bacteria, but that gives us sort of an idea that resistance is not inevitable. Teixobactin is now in advanced preclinical IND development. It was supposed to go into phase 1 this year, but because of COVID, it's not this year, but probably, hopefully, everything continues to be good. It will go into phase 1 trials next year. All right, so this experience with teixobactin was encouraging for us, and we thought that, we'll just continue mining uncultured bacteria and we'll start hitting upon gram-negative pathogens. And that didn't really happen. It didn't happen because the probability of finding any hit against gram-negative bacteria is at least 10 times lower than against gram-positive. That may not seem like a huge deal, but it took us about 10 years to get to teixobactin and then another backup lead that we have, right? So then given this hit rate, it will take us about 100 years to get to a gram-negative, anti-gram-negative compound using this approach. So I was thinking, how about shrinking that timeline to have a sort of a more realistic probability of finding something acting as gram-negative bacteria, and that's what I will tell you about. So here's our, standard drug discovery pipeline, pretty straightforward. So we looked at each and every element of that pipeline with the aim of resolving bottlenecks, and we've done that resolving in a number of cases, but today I'll tell you about only one for which we actually have proof of principle. So the largest bottleneck is also the one that is not discussed in the profession. And that is the bottleneck of the source. So whatever you do in natural product discovery, you start with a small sample of soil. You assume that that small sample of soil contains a producer of a wonderful new antibiotic, and then with whatever clever methods you try to find it. And that assumption, your initial assumption, is based on absolutely nothing. You have no idea whether you have a producer of a good compound or not in that soil sample. And since there's nothing to be done about it, there isn't much discussion of that initial bottleneck. So I submit to you that there is a straightforward theoretical solution of that problem. It's very simple. Instead of screening grams of soil here and there, you need to screen the biosphere. Now, there's a practical screening the biosphere. It is somewhat larger than what we can physically handle by 24 orders of magnitude. But there may be an approach to solving this problem and to closing the gap between these two different bodies. So let's assume that there is a group of bacteria that shares the requirements for antibiotics with us for whatever reason. So here's what we are looking for in an antibiotic. We want it to be active against gram-negative pathogens, have low toxicity, good PK, and in vivo efficacy. So if there is that group of bacteria, if it does exist, then it would have spent a number of millions of years collecting genes coding for these antibiotics from the biosphere by horizontal transmission of DNA. So what I'm saying is that the screen of the biosphere has been performed multiple times. And so what we need to do is to zero in on groups of bacteria that perform the screen the way we would want to do it, and tap into the results of that screen. So from this short list, this group of bacteria comes to mind. These are Xenorhabdus and Photorhabdus, which are symbionts of entomopathogenic nematodes. So they are part of the gut microbiome of nematodes. And when the nematode infects insect larvae, the nematodes release these Photorhabdus, and the Photorhabdus start making antimicrobial compounds to fend off competitors. Now, in that environment, the main competitors happen to be gram-negative bacteria. Compounds need to be of low toxicity, not to harm the nematode, which is a little animal. They have to have good PK. I think this is key. Good PK. So the ability to travel through the tissues of the larvae. That was kind of hard to zero in on. And of course, in vivo efficacy, they have to be efficacious in this insect larvae. So that's our short list. So here, a postdoc in my lab, UMI, started by screening a very small panel of Photorhabdus on a Petri dish overlaid with E. coli lawn. And here's your typical result. You put down a patch of Photorhabdus cells and nothing happens. Here is your positive control, Penibacillus polymyxa, that makes polymyx. And of course, this is what we want to see. This is what we're looking for. So then we started thinking, well, maybe the interesting stuff is sitting in the silent operons of Photorhabdus. And so if we take an extract, again, we don't see anything, but if we concentrate this extract 15-fold, now if you squint, you can see there is a very small zone of inhibition around 15-fold concentrated extract. And so based on that very small zone of inhibition, we isolated this compound, which is darobactin. It's a novel compound, represents a novel class of antibiotics with activity against gram-negative bacteria. Has an unusual scaffold, 2 linked rings. In order to make this one, for example, you need to make an unactivated carbon-carbon bond. That requires a free radical reaction. And indeed, the operon codes for DAR-E, which is a radical SAM enzyme capable of catalyzing such reactions. But this is unique for antibiotic, that carbon-carbon theme. Now, interestingly, this operon was borrowed by Photorhabdus. It has extremely low GC content. It came from a creature with a low GC content, and we have no idea what that is. We just know that this is clearly not an actinomycete, which have very high GC content. This group has been around for 370 million years, the Photorhabdus. Intuitively, it seems that that's been enough time to test DNA floating around the biosphere for genes that this group was interested in. So this compound has pretty good activity against the important number of important gram-negative pathogens like Klebsiella, E. coli, including the nasty multi-drug resistant versions of these. It has no activity against gram-positive bacteria and is inactive against gut symbionts and has no cytotoxicity. So overall, I'd say an interesting spectrum. has efficacy in a mouse thigh. That's the golden standard in anti-infectives. You want your compound to show efficacy in a mouse thigh infection. So here, darobactin acts a little bit better than gentamicin against polymyxin-resistant E. coli in this case. So we got resistant mutants to this compound. They all map to the same target, which is BAM-A. BAM-A is one of the two essential proteins of the outer membrane. It is a chaperone and insertase of outer membrane proteins. So it itself is a beta barrel, a typical fold for an outer membrane protein is beta barrel. It takes in incoming peptides, folds them into their own beta barrels, and makes things like outer membrane proteins porous. Now, there's been a serious effort to find by screening primarily hits against BAM-A, which did not really pan out. This is not a classical druggable target. It's not an enzyme. It doesn't have an active site where you can fit your small molecule and nicely and inhibit it. So that was very interesting. So how does darobactin inhibit BAM-A? Well, now we know how it does it. We've been collaborating with Sebastian Hiller and Handip Kaur and his group, co-crystallized darobactin with a target. So the paper actually just came out. It says accepted. Well, it came out this month. And so what darobactin does, it binds to the open lateral gate. So this lateral gate is where substrates, peptide substrates, BAM-A bind, forming a beta-beta strand. Now, the amazing thing about darobactin is that this backbone, this peptide backbone, is a preformed beta strand. So I thought that the warhead is going to be these two intricate fused rings. The fused rings, their only function is to rigidify this backbone. So usually you think about a peptide backbone as something standard and boring. Well, this is the warhead. This is a preformed beta strand that binds 1,000 times better to the open beta-1 strand gate than a natural peptide. So not surprisingly, this is how it jams the opening of the gate and prevents binding of native substrates. Now, an interesting thing about darobactins is that if you look computationally at bacterial genomes, it is present, this operon, the homologs of this operon are present in quite a number of bacteria, all of them gram-negative proteobacteria, and all of them live in association with animals. So that's sort of the general theme. These are in animal symbionts or pathogens. And that gives us a glimpse of Mother Nature doing medicinal chemistry, if you will. There's a bunch of analogs. We published these 5. We have now 11. And these we are evaluating for superior pharmacological qualities. Another interesting thing about darobactins is their behavior in vivo. Now, you can raise mutants to darobactin, which is an obvious liability. But we decided to test those mutants. These are E. coli mutants for virulence. And what we find is that here's your control. This is, mice die when infected with E. coli, wild type. But if you infect them with BAM-A mutants, these are avirulent. So it seems like these mutations cannot be acquired with impunity. There's a fitness cost to them. Now, whether all such mutations will carry a fitness cost, that's not clear, but certainly this is a good property to have for a drug because it diminishes the effective frequency of mutations considerably. So we're also thinking about a more general solution to this problem of resistance that we all have to struggle with. So the simple proposition is to use combinations. And of course, physicians today use combinations of compounds for a variety of reasons, including to diminish resistance development or to hit a particularly problematic infection. But all of these are combinations of compounds to which there is pre-existent resistance. And so we were wondering that a proper solution to this problem is to combine compounds for which there isn't any pre-existing resistance in the population. So here's darabactin, it's a novel compound, and we're combining it with epetraborole. Epetraborole is a terrific compound. It was in development by GlaxoSmithKline for a while. It acts against gram-negative bacteria, hits leucine tRNA synthase. But there was a prohibitively high frequency of resistance development that failed because of that in phase 2. So here's resistance to darabactin, here's resistance to epetraborole, and here's the frequency of resistance to the combination we cannot measure. It's below 10 to the minus 11. The theoretical frequency is 10 to the minus 15. I think this is a general recipe for combating antimicrobial resistance crisis. Figure out how to discover compounds and do not introduce them as singlets. If you want to beat our pathogen, introduce them as combinations. So with that, I thank my team, my collaborators and donors, and I thank you for listening.
Dr. Lewis, what a wonderful talk. I just thought it was fantastic. You know, you talk about combination therapy, but right away I think about antagonism. And, you know, that when you go to the patient model, if you will, when you get to clinical trials, you find that a lot of combinations have antagonism. And also, you know, in terms of automembrane biosynthesis inhibitors, you know, some of them have failed, correct me if I'm wrong, in terms of clinical trials. I just wanted to hear your opinions about that.
Sure, yeah. So yeah, of course, some compounds are going to be antagonistic. Now, there's one area, TB, where you only introduce combinations. All therapies, again, unfortunately, they still continue to introduce one at a time, right? Instead of at least 2 novel ones, which would be the way to go. So I'm waiting for them, you know, to do something more imaginative. But in that area, antagonism has not been a big problem. So even though we obviously need to avoid antagonistic, I don't think that will be a big obstacle. Like, for example, darabactin and epetraborole are not antagonistic. They're additive. Now, as to the failure of compounds against the BAM-A, you obviously are talking about the polyphore compounds.
Yeah.
Miripavidin. So those are polycations, and they hit not BAM-A directly, but they're chimeric compounds that hit LPS. So they are analogous to polymyxin, if you will, and also anchor BAM-A, right? So it's kind of a dual binding. But in order to bind to, because it's very hard to make a compound that's going to bind BAM-A unless you have something like darabactin that evolved to do that. So you first anchor the compound on LPS, and then there's some binding to BAM-A, and together that gives you a very potent compound. But the downside is, in order to target LPS, you need to have a polycationic structure. Polycations have a known liability, nephrotoxicity, and that's where that compound failed. So I think you need to avoid polycationic structures.
Thank you, Dr. Lewis.
Okay, Professor Lewis. A question from Nora Vasquez Laszlo. I messed her name up without a doubt. She wants to know, what was the logic behind combining darabactin specifically with epetraborole? Or was there a logic?
Yeah, that's a good question. You see, our options were extremely limited. what we were looking for a compound which is a realistic drug candidate, acts against gram-negative bacteria, and has not been approved and is available, right? So by those criteria, we found exactly one compound, the petrobor. So that's what was our choice.
Okay, thank you for that answer. I have a question or point. A number of years ago, a German friend of mine was looking at similar linear rigidified polycations as Alzheimer's treatment. Have you ever thought about that?
Well, as I mentioned, polycations have this, like, the moment you have more than 2 positive charges, you're in very big trouble.
Yeah, that I know.
Because of nephrotoxicity, so we try not to go there.
Yeah, is it not better to have trouble with nephrosis than to have Alzheimer's?
And to have Alzheimer's.
Yeah.
Yeah. Well, I don't work on Alzheimer's, so
no.
Okay. I wouldn't know that.
And if I can ask one more question, Dr. Lewis, you know, your example of succinate dehydrogenase with regards to your conclusion was, as you inactivate or have a reduction in these Krebs cycle proteins, that what you have is a reduction in overall ATP synthesis, correct? And
Yes.
This leads to the formation of persisters. You know, when I think about those proteins, they have a lot more effects than just ATP synthesis. Is there anything to my question there?
Yeah, well, of course, it's a very important question of causality. So one simple experiment that we performed is that you can add arsenate to cells, which causes a futile cycle of ATP depletion, right? So you get ADP arsenate spontaneously hydrolyzes. And so that's a way to specifically deplete ATP. You add arsenate, you get essentially full tolerance. So we knew that if you selectively drop ATP, that is sufficient for drug tolerance. And then for us, the question was, how does that happen in the cell? And of course, you know, if you don't have a Krebs cycle enzyme, that's going to drop your ATP. And we also show that directly by just knocking out the Krebs cycle enzyme. Of course, ATP, that's kind of a silly experiment, but you know, you have to do that.
Okay.
Thank you, Dr. Lewis.
One last question. It comes in from Sylvia Cardona. Could you explain or expand the concept of target-specific antibiotics and multi-target antibiotics? And they would like you to answer this question live. I'm not sure how you do that, by the way.
Multi-target, just something about multi-target antibiotics.
Okay, we have target-specific antibiotics and multi-target antibiotics.
Right, sure. Yeah, so the multi-target antibiotics are the major antibiotics in the clinic today. So that's fluoroquinolones that hit DNA gyrase and the homologous DNA topoisomerase, or the very many antibiotics such as aminoglycosides that act against the ribosome, but in fact they bind to 16S RNA, so some others bind to 23S RNA, and there are multiple gene copies of these RNAs. So that is in fact, even though they all bind to the same target, 16S, but since that is coded by different genes, in order to get a mutant, you need to have rare segregation of that mutant, right? That's equivalent to multi-targeting. And finally, you have beta-lactams, which bind to penicillin binding proteins, which are transglycosylase enzymes. And then you have a bunch of those, and they're all essential.
Okay.
Dr. If I could comment, I also think about antiseptics and disinfectants, that these things in general are multi-targeting. And also when you think about it, there's concentration dependent effects that also take place, considering that triclosan at low concentrations inhibits, fatty acid biosynthesis, but at high concentration, it acts as a bisphenol. And my suspicion is it denatures the heck out of everything, but just a comment.
No, of course, but there's a reason why we don't inject ourselves with triclosan, right? In the mouthwash.
Yeah,
So-
even when a certain person tells us to, so that's a good thing. At least bleach, anyways.
No, you should breathe bleach, not inject it. It's quite a different concept.
I'm not an expert on bleach, I must confess.
Yeah.
I am. Anyway, Dr. Lewis, I would like to thank you again for a wonderful conference, a wonderful talk, and thank you again.
Thank you all.
Okay, thank you. Well, we are at the end of our time. So,
If I could just make a comment, especially with these powerhouses of antimicrobial resistance in the room, it seems to me it's about time that we have a national center to get all you guys together in terms of, antimicrobial development. Hey, maybe even a world center. You keep thinking about sources of funding for things like that, but wow.
NDPI.
Yeah, there needs to be. I don't know if MTPI could fund something like that, but there's got to be some funding sources out there that would be interested in something like that. Comments?
There are very, very few worldwide sources of funding. You can get bilateral, maybe European, maybe North American. But then when you start going out and trying to get into the other sources of funding, so if you want to combine Japanese, Korean, Chinese, Indian funding, it's a non-trivial problem, which I will happily leave you to deal with as I'm retired.
Okay. Thank you.
Gus, I think there's some funding going on. Like I was funded for my PhD for treating multi-drug resistance.
But you were funded in Europe. You were not funded from Europe to do your PhD work in Hong Kong.
No, because it was a European
Gus is
thing.
asking.
Yeah, I guess so. But we had collaborations and we had partners outside of Europe. They were not as involved as the main people in the project, the PIs, but all the people.
It's the old adage, working in a vacuum, I suspect, is what I was trying to get at. And it seems to me that since this is a world issue, and we all know what's coming in terms of the potential of antimicrobial resistance overtaking the antibiotics that we have, that it seems that it would be a worthy thing to do in terms of governments actually getting today. And I guess, in our case, it means talking to Congress and talking to other individuals.
Ladies and gentlemen, I think at this point we should probably close down for the evening.
Yeah. Okay, I just want to add a few little things to the discussion before we're having about this global collaboration. And I think that's probably a good thing that we were going to take after this global situation that we're living now, that maybe for the next emergency, we will learn from this previous experience, hopefully.
Well,
how to collaborate between countries and multisectoral. What do you think, Gus?
Don't know how many others have ever done this, but I will speak from experience now. I have a project that we're currently getting to publish, which involves people who came from Canada, the States, Ireland, France, Poland, Saudi Arabia, and Japan. And the big problem was that we were trying to do it using webinars. It's hard work.
Yeah. Well,
anyway,
I think that's another long discussion. Well,
very.
I think it's time for concluding, isn't?
It's a conclusion. I would like to thank, in conclusion, all of today's speakers. It's been wonderful. I would like to thank you all very much for having actually sat down and given your talks in the time. If any of you have ever given talks in France, you would understand when I say that runovers are a horror. So anyway, I will shut down. I will turn it back to Cedric. And I thank you all very, very much.
I would also like to thank especially the session chairs and of course the speakers for accepting to take part in this second live session and for these interesting talks. Thanks to all participants for joining us today. Thank you very much for your attention. Have a nice day or evening wherever you are around the world. Goodbye and take care.
Okay, thank you. Bye-bye.
Thank you.
Bye.
Bye-bye.